So it's sometime on a Wednesday evening. A bunch of people took an exam this morning, and that's fine. And I just spent way too much time trying to improve the sound quality on my recordings. You know, all the good like microphones you might get to make everything sound better, they're all out of stock everywhere, and that's okay. And this microphone I use either picks up every sound or no sound. So that's how my day has been going, because who even thought that was important? Not you, I'm hoping. But here we are, looking at the bronchial tree, and you can imagine why it's called a tree. That's slang, but whatever. The idea is it has this kind of amazing branching pattern, and it has to, because what you have is a huge number of microscopic alveoli that are like the size of a tiny capillary. They're microscopic, they're super tiny. And every one of them has to be connected to an air pipe that somehow connects back to this trachea. And so the way a biological system is going to do that is by branching a bunch of times and getting smaller with each branch until at the last branching it's all microscopic, and that's what we see kind of here. They're color coded based on where they branched off of the bigger airways. And yeah, so this is basically showing this but color coded. So it's kind of cool. So the carina is the last thing before the bronchi, and what we'll see is that we can describe different parts of the bronchi based on how branched they are and where they are. So the primary bronchi are these two things. These are the main ones that go into the lungs, and then very soon after they go into the lungs, they split, and a different branch goes to each lobe of the lungs, and so those are lobar or secondary bronchi. After that, they branch and branch and branch. And so the segmental would be visible here. They would be what's branching just after, just like here and here. So these would be secondary or lobar, and then these would be segmental. This becomes kind of silly because what we could say is there's one order of branching from trachea to two bronchi. The second order would be from primary bronchi to secondary or lobar. Third order branching would be the different segmental bronchi. Every order of branching makes smaller branches, and they can branch up to 23 times. And so hopefully that's giving you a sense of just how small these have to be when they actually supply an alveolus with air. So when they get smaller than a millimeter, which at this level it'd be visible, like that would be a millimeter. No, it might be smaller than that, but you can definitely see something a millimeter thick. When they're smaller than that, they're bronchioles, and part of why they get a different name, even though they're still maybe visible, is that the physical properties of something that's small happen to be a little bit different. So they can't be designed exactly the same way that something bigger could be designed, as we will see. So here's the idea. We are trying to get air into these little spheres. These spheres can expand. That's one thing they can do, and that is what they do when our lungs expand and contract. And they're surrounded by blood vessels or capillaries. And that allows them to do the external respiration that they do. So as we go from the main bronchus, which would be way up above here, because this would all be microscopic structures. As we go from the main bronchus down, we see a change in connective tissue. So the kind of regular cartilage rings we saw would become, in a smaller structure, less regular. And when we get down to bronchioles, they're really too small for cartilage. Any piece of cartilage big enough to be like recognizable as cartilage would have no flexibility at all. So you might as well use bone or rocks or something. So at a much smaller level, you can use proteins to get the same kind of strength that you would need something stiffer to do at a larger size, like cartilage or bone. And then the epithelium also changes. It goes from the respiratory mucosa, that PSCC. What does that stand for? Remind me what that stands for. 3, 2, 1, pause. To the different simpler types of epithelium, and the last one you would get is simple cuboidal. And then, yeah, the other thing would be the smooth muscle. So at the size of the trachea, you have skeletal muscle. You have that controllable trachealis muscle. But when you get to the level of the bronchi and the bronchioles, you no longer have voluntary control over that. And smooth muscle at something this size can be the only structure or the most dominant structure, and it can also cause bronchoconstriction and bronchodilation. Let's see. You might have looked at a slide like this early in the semester. I really don't remember. Yeah, I don't remember which ones you saw and which ones you didn't see. But this would be a cross-section through a lung where each of these things I'm pointing at would be an alveolus. And these cells around the outside would be simple squamous alveolar cells. And yeah, so this is what we have. The respiratory bronchioles become alveolar ducts, which would really be too small to see, and those would feed alveolar sacs. And so what they're showing is in this cross section, a respiratory bronchiole would split into alveolar ducts, and those would have alveoli connected to them. So the air, I mean, this is not a three-dimensional structure we're seeing. This is a cross section of a three-dimensional structure, but air would come in this way and split off and then go into all these different alveoli. That's the plan. Okay, so the alveoli is the respiratory zone. This is the region where the gas exchange or the internal respiration happens. And so this is, the alveoli are the lungs. They are the reason the lungs exist, and they are what we try to protect. Why we have that respiratory mucosa with the mucus that gets trafficked to the stomach. It's to protect these guys. These have a great blood supply, and they're kept moist and warm, and that is very appealing to microorganisms. So the fungi we inhale, the bacteria we inhale, the viruses we inhale, they would all love to infect our alveolar cells and get into the bloodstream through there. And so our respiratory system tries to prevent that. And yeah, so let's get into it. This is a picture you're going to see several more times on the next few slides, and what it's showing is it's trying to show a three-dimensional representation of like half of an alveolus here. It's also showing the fact that the alveoli are connected to each other. In some cases, they will share epithelial cells. It's also showing that they're only one epithelial cell thick, and it's a squamous cell that is very narrow, or very thin, I should say. And what that means is that gases can diffuse across it easily. And what they're trying to do is get the gases that flow in here. If it's oxygen, it can diffuse into the bloodstream, into the bloodstream, into the bloodstream. So into these capillaries, and carbon dioxide will diffuse into the alveolus from the blood. What causes that diffusion? I mean, what is the physical process that pushes those molecules? You tell me. 3, 2, 1, pause. I'm hoping what you came up with was diffusion is random motion of molecules, because that really is what it is. It's random motion, but if you have a high concentration of carbon dioxide here and a low concentration of carbon dioxide here, random motion of carbon dioxide is going to leave a lot of it here. That motion will tend, on average, to put the molecules here until the concentration here and here are the same. So what diffusion could not do would be to pump all the carbon dioxide out of the blood. Diffusion cannot pump all the oxygen into the blood. What we're always getting is some fraction of the carbon dioxide is taken out, some fraction of the oxygen is put in. And that's an important thing to remember. And when you tackle this stuff in physiology, what you'll see is that blood has a lot of oxygen in it, always. When we say it's deoxygenated, it still has a lot of oxygen. But it has more carbon dioxide. I mean, imagine you can do CPR on a person. You can exhale into another person's lungs and provide them enough oxygen to survive. Because your lungs are not taking all the oxygen out of the air, and when you exhale, you're not taking all the carbon dioxide out of the blood. So I focus on this kind of topic because when you study microscopic things like the bacteria that I used to study, or if you study, I don't know, processes like the engineering I used to study, you can't think of any process as being 100%. So you'll enjoy that when you get to the next class you take. Okay, so that's a lot of stuff I just told you about this, but what these slides are trying to show you with this diagram is the different pictures of the different types of cells. So the type 1 cells, that's kind of a dumb name, but what they do is they are the cells that make the walls of the alveoli. So this is a type 1 cell, and all of these like tan cells you're seeing are type 1 cells. And here's another type 1 cell, and here's another type 1 cell. They make up the structure, and they're thin, and so a type 1 cell can be welded to a squamous cell from the outside of a capillary, and it's easy for gases to just diffuse across those membranes. Yeah, so they share a basal lamina, and remember the basal lamina is just a thin protein layer. Yeah, and I guess, okay, yeah, so this has its own name, the respiratory membrane. Cool. The next type of cell, I would show you the type 2 cells, which again, stupid name, what are you going to do? The type 2 cells here are, let's see, yeah, they're the green ones. And their job is to make a surfactant. It's a fluid that I believe is called surfactant, and it is also an example of a surfactant. And that's, a surfactant is a chemical that can reduce interfacial tension, or in this case, surface tension between air and water, or between water and water. And the idea is the internal surface of an alveolus is wet. So it has water, water, I guess, because it's been humidified. And since the cells are wet, if they were to touch each other, they would be stuck together if it was normal water. And in that extreme example, yeah, if this collapsed to the point where this cell touched this cell, they would be like welded together, just from surface tension of water. To prevent that, you could add a detergent to the water, or a surfactant, that would reduce the tension of water and it would let those things separate. That's kind of the extreme example of what surfactant would do. If this cell and this cell touched and the layer of water welded them together, surfactant would kind of disrupt that and let them separate again. More realistically, what's happening is as these contract, just the water tension is going to make it harder for them to expand. And so surfactant kind of makes it easier for them to expand. So that's what the type 2 cells do, is they make it easier for these to expand, just by counteracting the effects of water surface tension, or the interfacial tension between water and air. And then the last type of cell in the respiratory zone would be the macrophages. These are cells from the immune system that their job is to do phagocytosis of foreign material. So they would tackle dead cells. So our cells, when they're old and dead, macrophages will kind of recycle them. But also when they find bacteria, mold, dust particles, anything that made it down to the alveoli that doesn't belong there, a macrophage will eat. And if you, I probably already told you this, but if you were to look in a sample of a lung of someone from Southern California, one weird thing you see is macrophages with little black dots in them. And what it turns out in Southern California, one thing we all have in common is that we are always inhaling microscopic bits of rubber from tires. And nothing in our body can break down tire rubber. So our macrophages kind of gather it up and they keep it within themselves, but they can't do anything with it. So over time, someone in Southern California accumulates rubber in their lungs, which is wild. What macrophages are really supposed to do is, once they pick up things like bacteria or tire rubber, they migrate up through the bronchi to get to, ultimately to get to the larynx and then go down the stomach and out the body. Because if we can't break down tire rubber, we can send it through our digestive system and poop it out. No problem. But that won't always work. So if you're looking for some rubber, look deep within you. Okay, the next thing would be, I mentioned this before, how the pleural cavities are defined with this parietal pleura, which is kind of welded to the external, to the wall of the thoracic cavity, and the visceral pleura is a thin layer that's sort of welded to the outside of the lung, and in between them is a liquid called the serous fluid. And so that this is how you should picture it. There's the parietal pleura is attached to the external surface, the visceral pleura is attached to the lung, and there's that liquid between them. So when you do something like contract your diaphragm and pull this down, it's going to pull down on the surface of the lung. But how? What is it grabbing to pull on the lung? What is it attached to? All it's attached to is the parietal pleura. All it's attached to is that one layer of that one serous membrane. And the only thing attaching that layer of the serous membrane to the visceral layer of the serous membrane and your lung is that liquid. So that liquid is slippery and it's going to reduce friction, but also it uses its own surface tension to make sure that these two layers never separate from each other. Now, if we disrupt this, so if you poke something through a lung, you can introduce air into that space between them and the lung will collapse. The person won't be able to inflate their lungs anymore because no matter how much they expand their thoracic cage, nothing is pulling on the lung anymore. So the condition of pneumothorax, or collapsed lung, happens whenever air can get in between those two layers of the pleurae. Into the pleural cavity, so to speak. So that's wild. This is one of my favorite diagrams from this class because it shows you things you know. It shows you structures you have learned. Everything on here you should be able to label, but it's tricky at first. it's always hard to look at a new section from the body and identify the structures, no matter how well you learn them. So I do recommend you pause and look through it, and when you study, cover up these labels and relabel them. But anyway, what this is showing, if we take this transverse section of a person, is that where the bronchi enter the lungs is right next to where the pulmonary arteries enter and the pulmonary veins exit. And they're all going through that one spot called the hilum, or actually hilus. You could see hilus or hilum. I think this is the more modern word. And this is what it would look like if you cut the tubes. And this allows us to see the right lung and the left lung and the different lobes. So in each case you have a superior lobe, and you'll have an inferior lobe, but also the right one has a middle lobe. The left lung doesn't have that extra lobe. Instead, it has an indentation for the heart called the cardiac impression and the cardiac notch. How cool is that? Okay. Yes, and up here, apex, down here, the base. This is a lung from a cadaver that has had wax put into the tubes to show you pulmonary veins and pulmonary artery, and then the bronchus. Oh, and then this is a cadaveric image of lungs. They have collapsed because they have disconnected from from the thoracic wall. And so what you'd be able to see here, the heart would be here inside the pericardium, and you'd have sort of this cardiac notch, which is a place where the lung just isn't going to prevent the heart from expanding, I guess you could say. And then the lobes are separated by fissures. So you have 3 separate lobes over here, 2 lobes over here, and they're separated by fissures. This would be a review of the parts of the respiratory system. Boom. Okay, so that's the respiratory system. The other slides here are a review of the way breathing works and the muscles that you use. And if you're like most anatomy students, when you look at muscles after the muscle exam, it gives you a feeling of, comfort to see something that you don't have to memorize. So if you didn't feel that way when you saw the muscles, cultivate that feeling. And I will see you when we reconnect over the lymphatic system.